All right, we've had a look at how we can amplify DNA using polymerase chain reaction, so create hundreds and hundreds of copies of the same sequence of DNA from a small sample. Now we're going to look at the concept of actually sequencing DNA using electrophoresis, and how we can then actually go about not only using electrophoresis as a process, but being able to then do processes to enable us to actually sequence the DNA. So find out what is that ATGC sort of sequence in particular orders. So the first part of this is gel electrophoresis, and this concept here is relatively simple in its essence. And what it does is it separates DNA samples based on size once we've cut them up. And so in this, what happens is we get a sample of DNA and we use restriction enzymes. We chuck them into the sample. And because restriction enzymes, like all enzymes, are unique to substrates, they will cut at specific parts of the DNA sequence. And so they effectively chop up DNA based on the specific enzyme that it is and where it actually meets the substrate that it needs to actually enact a change for. So then what we have is we have this sort of pool of all different size segments of DNA. And what we can do is we can chuck it into a gel electrophoresis machine where we chuck samples into what we call wells. And because we know that DNA has a negative charge overall, if we put a current through that, um, what happens is DNA will move across the machine towards the positive charge. It's attracted towards that positive charge. The beauty of this as well is depending on where the fragments have been cut up, would indicate their size because, as we said, restriction enzymes, like all enzymes, are specific, so they'll cut at specific points. And so lengths of DNA that are bigger won't travel as far because they get caught up at the start of the machine, and smaller lengths of DNA that have been cut up by the restriction enzymes will move further across the machine. And so what does this look like? Well, what you can see here is a very simple representation of one of these machines. We have a buffer solution on either end, and what that does is that regulates things like pH, which we always need to be able to do. And we have wells at the start, which exist within what we call a gel, sort of like a gel layer, which is an agar sort of solution. And so we've put our restriction enzymes into our little samples of DNA, and then that's all cut up. That's done independently. We don't know where it's cut, but we know it's cut to unique sequences of DNA. And then those wells are at the negative end of the gel electrophoresis machine. And then what happens is as we turn the power source on, we get an electrical current move through, and the DNA actually starts to be attracted towards the positive end. And so as the DNA moves across, such as that, if it is smaller, it is able to move further along that gel. And if it is bigger, i.e., contains more nucleotides in that sequence, it doesn't travel as far in across the well. And so in that process, in its essence, gel electrophoresis will separate fragments of DNA that have been cut up based on their size. And what can we do with that originally? Well, without us knowing the specific sequence, it's still useful to know the size of where it cuts because we can compare samples from unknown things like crime scenes to samples that we obtain. And if we've got the same restriction enzymes, they'll cut at the same base sequence. So a sequence of, say, 800 nucleotides in a blood splatter will be the same from the witness or the person that we are sort of taking the sample from as well, because they have the same sequence of DNA. So it will cut at the same points, and so it will work or move in the same way theoretically as we put it through the gel electrophoresis machine. But we're going to focus on this picture on the right here for how we can look at that. And so this marker that you see on the side there, that allows us to indicate roughly if it moves, say, this far from the top downwards, we know that it's 1200 base pairs long. So we know that the sequence that exists within that has been cut by the restriction enzymes is, this long. It's 1200 base pairs. We know right down the very bottom, there is one that is about 100 base pairs. And so we sort of can get a gauge of length of base pairs that have been cut by the restriction enzymes from the marker. Now, when you chuck the 3 samples in the 3 different wells, so they might be from 3 different people, what we can see is we can see that in person A, for example, they have a sequence that was cut at 1,000 base pairs. In person B, we have an 800 base pairs sequence that was cut at the same marker and a 200 base pairs that was cut at the marker. And then in C, we have that sequence cut at 600 base pairs, a 300 base pairs one, and the 100 base pairs. So there's 3 parts of those samples. And if we had a sample D there and we saw that it was very similar to sample C, we could infer that maybe they were from the same person. And so gel electrophoresis, in essence, we use restriction enzymes to cut DNA based on size. We don't know where it's going to cut. We just know restriction enzymes are unique because they are an enzyme. And so what happens is they cut based on size, and then we rely on the understanding that DNA is negatively charged, and if we chuck it into a well, it'll move to a positive end if we put a current through it. We also need to know that we have a buffer solution, so a solution where we can actually have adequate pH in an environment, and we can have the gel ready to go so we can move it across when we put the current through. Absolutely watch this video. It probably will explain it in a much better way than I have. So the next part of this is actually, we've just talked about only separating DNA based on size, but what we actually want to be able to do is understand the sequence of DNA that exists. So we want to be able to map the genome, or we want to be able to understand what exists within those 1,000 base pairs, because that might make up the key ingredients for what that protein is that it does. And so DNA sequencing is a key process in understanding that whole sequence. And we sort of were able to really get this, completed for the human genome in about 2003. So a cost of about $3 billion was undertaken, and this took many years to be able to sequence the whole of the human genome. That's really, really important for us, because then we can understand where, what we're made up of, how we are consistent with our ancestors, how we compare to other organisms and things like that, where mutations exist and what they do to individuals and things like that. We can do this much, much cheaper now, and technology, as always, advances at a much more rapid pace than what it would have done 20 years ago. I'm going to skip past that. But the purpose of sequencing, creating things like personalized medicine, so developing drugs that are able to respond to your body, because my body is going to be different to yours. And so we might have minute changes that we need to create personalized medicine for. That would be fantastic. Then we could be ensured that when we delivered personalized medicine, it would actually have the desired response that we want. Studying relationships, like I spoke about before, the commonalities between us and other organisms, and looking at forensics, so understanding things like paternity testing, DNA profiling, that kind of stuff. All right, so this next part of this is where it gets a little bit more complex, but I'll try and make it as simple as possible. And so for us to be able to sequence DNA, something that we can't theoretically see, what we've been able to work out is that we can actually create modified nucleotides. And then, because we know DNA base pairing is universal, we can use these modified nucleotides to actually create DNA sequences that have a fluorescent marker on them as such. And so what happens is, we've used PCR, we've replicated strands of DNA, and then what we do is we actually go through replication processes. And so we do this in a lab where we heat up DNA samples, and we add primers in. And then what happens is we add modified nucleotides into a soup. And so we add one at a time. And what they do, these modified nucleotides, is they don't enable for further assembly of DNA like what would normally occur. So that would normally occur until we hit stop points in DNA sequences for where we find proteins. But when we add a modified nucleotide into the soup, if that is chosen and added to the sequence, no more base pairing can occur. And so what happens is we do this over and over and over again, and we add different modified nucleotides each time. And what happens is, as the DNA is being replicated, so as the A is being assembled against a complementary base T, C, G, and so on and so forth, if a modified nucleotide is picked up because we've added that one into the soup, it will then base pair there. That will stop and terminate the sequence. And so we get what you can see there is in the first sample, we've got a DDT. And so every time a T nucleotide was due to be replaced against the A, it instead gets replaced with this modified fluorescent DDT nucleotide. And so every time there's a T in the sequence, what happens is we might pick up one of the normal nucleotides, or we might pick up one of the modified nucleotides. And if we pick up one of the modified nucleotides, it'll terminate the sequence. And so what we end up with is we end up with all of these sequences of DNA where all of the T's eventually become mapped. And the same process happens with the A's and the G's and the C's. So then when we chuck those through those gel electrophoresis machines, what we can actually do is we can actually then go, oh, if this is, we know this has got a T terminating sequence. And so when that's chucked in, if that moves all the way at the end, we know that there's a T sequence furthest away, like that's the last point in that particular sequence of DNA. Whereas if it moves right at the very start, we might know that that's actually at a different point along that strand of DNA. And so what it allows us to do is it allows us to sequence DNA, but also allows us to actually work out whether there are things like mutations that exist there as well. And so probably this picture is not going to be the best one, but the next one is this one, and the following slides are the ones that we use this process for. So I guess understand from the previous slides that we add modified nucleotides when we do replication processes, and that enables us to find out where specific nucleotides are. And then because we can separate by size, we can then organize those in such a way where we can actually find out the T's are here, the A's are here, the G's and the C's are there. And so what happens is we run this through gel electrophoresis and we get what is called a electropherogram that is produced. And that is that picture on the very right-hand side there. And you can see it has all those peaks. And so for you to be able to understand that, it sort of just tells us basically along the base sequences, how many times did we see this particular sequence of DNA fluorescent because of the DD marker that was put in, so the DD nucleotide. And so here, at every time we ran it through the system, you can see at the start of this graph, at the 65 point and just before that, every single time, what we noticed was there was a terminating DDT marker there. And that always happened there. And at the next one, so the 66, for example, that moved a little bit further along the gel electrophoresis machine. So we able to say that was the next base in the sequence, and that had the terminating A nucleotide there. And so what you can see is that as this is run through, every single time it was a T, and then it was another T, and then it was an A, and then another A. The height of those peaks, or how far they rise up and down the graph, is how frequently we saw that being that particular code. What you need to understand is when we cut up DNA samples, we're cutting up hundreds and hundreds and hundreds of samples. And so when we run that through the machines, we're actually seeing that occur so many times. And so, for example, here, you can see at the 120 point, so where they're at the 120 base pair length, we've got that A that has the strongest indication that there is an A there. And so where you see ones that are much, much lower, and you might see some overlap, which we'll have a look at in a couple of slides' time, what that might indicate for us is that at that sort of point, there may have been a mutation in some of the DNA sequences within the actual pool that we saw. And if there is, then the signal strength's not going to be as strong. We're not going to see as many of that particular base being represented, and we might see a different base being represented. And what you can see here is in that top image, you can see that you've got some pretty strong signals. So you've got a pretty good indication of the sequence in its correct order. And when you can see when we undergo mutations and when we put those into the well, you can see pretty much half of the actual samples actually had a terminating nucleotide difference, where G and C were different. And so that's where we can see mutations occurring. Um, and that's when we're not as confident in what is the true representation of that sample. So we're not sure whether it is, a mutated sample or not. But essentially, you need to be able to read these, and you read them when you're given a key. And so the key will be provided to you if you are having to read them. It'll be color indicated, and then essentially you just have to go through and go, what is the sequence of DNA here? So in that top one, we would see that the sequence is A, C, T, T, C, T, T, so on and so forth. That's well and good when the sample is really clearly indicated. But when you get to things like the third sample, because there's been a lot of mutations that have occurred at that particular point, we're seeing a lot of overlap, so we can't be confident in the DNA sequence itself. And that's electrophoresis and electropherograms.